Okay, you can start now, Dr. Mohammed. You can start your lecture. Dr. Mohammed?
Yes, now I am here. Okay, thank you very much, doctor. Can I start my lecture? Can I start?
Yes, you can. You can.
Okay, everything is okay. Do you see my presentation, my PowerPoint?
Yes, it is clear, and your sound is clear now. You can start it now, Doctor.
Okay, thank you very much. Bismillah ar-Rahman ar-Rahim. Salamun alaikum. First of all, I should appreciate for your attendance in my lecture, in my presentation. And as you can see, my presentation, my lecture, the title of my lecture is Molecular Screening in Food Science, Molecular Diagnosis of Lactic Acid Bacteria in Dairy Products with Focus on Cheese. Let's introduce myself. My name is Mohammad Reza Edalatian Dovom, Associate Professor in Food Science and Technology Department, Ferdowsi University of Mashhad. And now I'm here to give my presentation, to give my lecture. I'm pleased to give my lecture for students of Basrah University, and it is my pleasure to give my lecture for you. Thank you very much. Okay, let's start for the molecular identification and screening. In order to molecular identification of lactic acid bacteria and also every microorganisms in foods, especially in dairy products like cheese, we have two kinds of molecular techniques. We can categorize and we can divide molecular identification into two subcategories, two subdivision, as you can see here. The first is culture-dependent molecular identification and screening, and the second one is culture-independent molecular identification and screening. As you can see here in this picture, shows you the sequences and the steps of identification of microorganisms from a sample, and the sample here is a soil sample, for example, and after DNA extraction, it ends to the 16S molecular typing and MALDI-TOF identification. And during these steps, we should isolate and extract the DNA from the bacteria, from the isolate, from the colonies. And after DNA extraction, we should perform some molecular techniques like PCR, I mean polymerase chain reaction. And finally, we can identify the microorganisms in a sample. For example, here in a soil sample. As you can see in this picture shows you the sequences and the steps of the molecular identification and molecular techniques. After DNA extraction, as I told you, we have the PCR, and this is the thermocycler of PCR, polymerase chain reaction. After that, we should perform the gel electrophoresis, and here we have agarose gel electrophoresis, as you can see in this picture. In the next step, we have a UV visualization. We should visualize the DNA band with the help of UV, ultraviolet. And finally, we have this picture, and this picture is the gel picture of the agarose gel. And here we have different bands, which is represents for every isolate or for every bacteria. Okay. Let's start with the first molecular techniques, and here I mean culture-dependent molecular identification and screening. For culture-dependent molecular identification, first of all, we should extract the DNA from the colony, from the microorganism or bacteria isolated from culture media, from the plate, I mean. So the first step is the DNA extraction from colony isolated from culture media or plate. In the second step, we should perform the PCR with thermocycler PCR of 16S rDNA from the colonies. As you know, the 16S rDNA is a gene which we choose, which we select because it has the conserved region and also some variable regions and also some conserved regions. So we should use and we should select this gene in order to amplify with the PCR and in order to identification. In the next step, after the PCR, we have ribotyping and amplified rDNA restriction analysis, or RDRA. RDRA is the abbreviation for this technique. This technique is a kind of typing techniques. In the next step, we have sequencing or sending the PCR product for sequence to the sequencing centers. For example, MWG in Germany or Isogen in Netherlands, or for example, Macrogene in South Korea. We should send our PCR products in order to sequencing to these centers. In the next step, we have identification by sequence comparison. And finally, we have Rapid Pcr typing. I will explain in more detail in the next slides for you step by step. In the next slide, I want to explain the steps of culture-independent molecular identification and screening. As you can see here, I divided this category into some subcategories here. In first step, we should isolate the total DNA from food sample, like milk, cheese, and so on. As you can see here, we should isolate the DNA from the food sample, not from the colonies. But in the previous slide, in the previous method, I mean culture-dependent molecular technique, we should isolate DNA from the colony, from the microorganism or bacteria. But here, we should isolate the total DNA from a food sample, like milk, cheese, and other food sample. In the second step, we should perform the PCR again, PCR of 16S rDNA gene. In the next step, we should perform denaturating gradient gel electrophoresis PCR. It is the abbreviation of that is Dgge PCR. Dgge stands for denaturating gradient gel electrophoresis PCR. In the next step, isolation of DNA from bands. And again, we have sequencing. And finally, identification by sequence comparison. I will explain more details in the next slide for you. Okay. In this slide, as you can see, we have a picture and we have two procedures or two methods for identification. For example, if we have a food sample like a cheese sample, as you can see here, it is known as Lyravon and Crusade. Let's explain more about these two kinds of cheeses. These two kinds of cheeses is the most popular and most important traditional and local dairy products and traditional cheese in Iran, which is produced in north east of Iran. It is called Lyravon cheese and Crusade cheese. So these two kinds of cheeses are the most important and most popular traditional and local cheeses which is produced in Iran. So here we have a culturing. Culturing means culture dependent molecular techniques, which starts with dilutions, after that plating in some kind of selective or elective media for lactic acid bacteria, because here we focus on lactic acid bacteria and we want to isolate lactic acid bacteria from the cheese samples. So we should use selective media for lactic acid bacteria like M17, MRS, KAA, and MRS vancomycin.
Excuse me, Dr. Mohammed.
Yes, yes, please.
If you can't change your slide.
Sorry?
Your slide, you can't change from your keyboard. Your slide.
My slide, yes. What happened for my slide?
Yes, now, yes, yes.
And now it is okay?
Yes, yes. Thank you.
Do you have any problem or no?
No, no, only change. When you explain which slides you change on it.
Okay. Now we are in the eighth number of the slide.
Okay.
Is 8.
It is now.
It is okay.
Yeah.
It is okay.
Okay.
Okay. Let's continue. Yes, let's continue. So after plating, we have a PCR of 16S rDNA from colonies. Here, we should extract the DNA from the colonies before the PCR. Before the PCR, we should perform DNA extraction. We should extract the DNA from the colonies. Here, colonies means the colonies of lactic acid bacteria. So in the next step, we have the Rrna. Rrna, as I mentioned for you in previous slides, it's a kind of typing and differentiation techniques, molecular techniques. And after Rrna, we have a sequencing step. In this step, we should send the PCR products to the sequencing centers, as I told you before. And after sequencing, in the next step, we have identification by sequence comparison. And in the final step, we have Rapid Pcr typing. These are includes the culturing or culture-dependent molecular technique. So let's go to the second procedure in the right hand, as you can see. PCR Dgge. PCR Dgge is a kind of culture independent molecular techniques, because here we isolate the DNA or total DNA directly from a food sample, not from the colonies. As you can see and as you can compare these two methods and these two procedures here, in the culturing method, we should extract the DNA from the colonies, which is produced on the plate, on the media. But in the second procedure, in the Dgge, we should isolate the total DNA directly from a food sample, like such as Lyravon or Crusade cheese. And here we have, for example, 2 examples from a cheese. So after isolation of total DNA from a food sample, we should perform the PCR of 16S rDNA. And after PCR, we should perform the Dgge, and I will explain more about the Dgge in the next slides. After Dgge, isolation of DNA from bands, and after that we have a sequencing, and finally we have identification by sequence comparison, as you can see here. So you can compare here between these 2 methods. As you can see, these 2 procedures have some similarities and have some differences. Okay. And here, let's start with the Dgge analysis of traditional cheeses like Lyravon and Crusade, as I told you. And as I mentioned for you, these kind of cheeses are the most important and most popular traditional and local cheeses which is produced in Iran. In the first step, we have the extraction of total microbial DNA directly from the food sample. In order to extract the DNA, first of all, we should thawing or defrosting the frozen samples, because the samples before have stored at -18 at freezer, I mean. And first of all, we should thaw or defrost the frozen samples at 4 degrees centigrade at refrigerator, I mean. After thawing, we should homogenize the sample, the food sample, in 2% sodium citrate. And after that, using homogenate for isolation of total microbial DNA. And finally, we can measure, we have measurement, the DNA quantity and quality by absorption at 260 and 280 nanometers using the nanodrop spectrophotometer. As you can know, this picture shows a nanodrop spectrophotometer, and nanodrop is used for measuring the quantity and quality of the DNA, of the extracted DNA, and we can measure the quantity and quality and purity of the DNA at these 2 wavelengths, I mean 260 and 280 nanometers, with the help of nanodrop spectrophotometer, and as you can see in this picture. And also in the next slide and next picture, you can see here again a nanodrop spectrophotometer. And here is a curve which is produced with the nanodrop spectrophotometer here. And here you can see the ratio of 260 to 280. For example, if this ratio more than 1.6 is okay, the quality and the purity of DNA is okay. And also here you can see the concentration and the quantity of the DNA, of the extracted DNA. For example, here the concentration of extracted DNA is 6 to 5.4 nanogram per microliter. And here it shows that the quantity and the concentration of DNA is okay and is good, and you can use this DNA for PCR. So in the next step and in the next slide, we can start with the PCR amplification of 16S rRNA sequences. As I told you before, we choose and we select the 16S rRNA gene because it is the most suitable gene in order to amplification and in order to identification of the bacteria, especially lactic acid bacteria, and also every prokaryotics. So we should amplify 16S rRNA gene after DNA extraction. In order in order to PCR amplification, first of all, we should select or collect 100 nanogram of purified DNA was used as a template in PCR amplification. This is the amount, or this is the concentration or amount of the DNA which we can use for PCR. And for PCR amplification of V3 region of bacterial 16S rRNA gene using universal primers, as you know, in order to perform the PCR, we require universal primers, and here these are the universal primers F357 GC clamp, and this is the sequence of the primer, of the forward primer with a GC clamp, and for another one is a reverse primer, and here reverse primer is R518 or 518, and this is the sequence of this primer from 5' to 3', and these primers are published in the papers. In some papers, you can
please only slide, change your slides.
Change my slide. Do you have any problem?
Yes.
Okay.
Yes.
Can you see? Yes.
Yes.
Can you see? Can you see?
Yes. But you, every time you explain another slide, you change. You must change. Because your slides.
Now, can you see my slide? Can you see my slide? No problem.
Yes, of course, no problem.
Okay. Let's go to the next. Let's go to the next slide. In the next slide, it shows the amplification of the D1 domain of the 26S rRNA gene of fungi. In order to amplification for the fungi or eukaryotic cell, you should use another region or another gene, and here this is the gene 26S rRNA gene of the fungi, because these are the eukaryotic. And here we have two different primers, another primers, I mean using primers NL1 GC and LS2 GC. As you can see, LS2, these are the forward and reverse primers in order to amplification of D1 domain of 26S rRNA gene of fungi. Okay, in the next slide, as you can see here, we have the electrophoretic conditions. After doing and after performing the PCR, we should perform the electrophoresis. And so we should know the electrophoretic conditions here. In order to perform the Dgge PCR in a decode apparatus or biorad apparatus, we should use, first of all, 8% polyacrylamide gels. We should prepare, we should make a polyacrylamide gel with the concentration of 8% polyacrylamide gels. The temperature should be adjusted at 60 degrees centigrade. And the best separation of PCR ampli-cones at the denaturating gradient of 80 to 60% for bacteria and 60 to 50% for fungi. As you can see here, the concentration of denaturating gradient is different for bacteria and for fungi. And for the electrophoresis conditions, we have here at 75 voltage for 16 hours for bacteria. We should adjust this volt or voltage, and this time for bacterial, and 130 voltage for 4.5 hours for fungal amplification. And we should adjust the apparatus, the equipment of electrophoresis at these voltage and at these times. And finally, bands were visualized under UV light, under ultraviolet light, after staining with ethidium bromide and photographed. And finally, we can see the picture of the bands in the gel. In the next step, identification of Dgge bands. After appear the bands in the gels, we should identify the Dgge bands, and in order to identify the Dgge bands, assigning to species by comparison with a control ladder of known strains. We should compare every band with a control, I mean like a positive control ladder of known strains, which I will explain for you in the next slides. So here, almost the first identification, I mean the independent identification is finished in this slide. And in the next slide, let's start with the second approach. And the second approach is culture-dependent approach. Now I want to explain for you the culture-dependent approach. So in order to molecular identification with culture-dependent approach, first of all, we should cultivate the LAB here. LAB stands for lactic acid bacteria, because the purpose and the goal of our research is the identification of lactic acid bacteria here. So we should cultivate the lactic acid bacteria in a corresponding media like M17 or MRS. In the next step, we have molecular identification of LAB species. In order to molecular identification of lactic acid bacteria, we have a polyphasic molecular procedure. First of all, we should extract the total DNA. We should extraction of total DNA from the colonies, I mean lactic acid bacteria colonies, which is produced, which is have growth in a media. After that, partial amplification of 16S rRNA genes. In the third step, we have amplified ribosomal DNA restriction analysis, or ARDRA. And finally, we have sequencing and sequence comparison with some databases. As you know, we have some databases in bioinformatics like NCBI or GenBank. And finally, we should make comparison or compare or results or sequencing with the sequences which is deposited in the some databases like NCBI or GenBank. Okay. In the next slide, in this slide, I want to explain for you the first step of culture-dependent approach. In order to start the culture-dependent approach, first of all, we should have a DNA extraction. And in order to DNA extract from the colonies, we have two methods, I mean two procedures. First of all, with the thermal processing here, for example, with the thermocycler here, as you can see, this picture is related to the thermocycler, and we can extract the DNA with the thermocycler. And the second method, DNA extraction with the mini bead beater and glass bead, and this is the picture of mini bead beater apparatus or equipment. So As you can see in the next slide, the DNA extraction. Here we have two procedures or two methods. In the first method, we can heat at 98 degrees centigrade for 10 minutes in a thermocycler, as I showed you in the previous slide. And after that, centrifuge for 5 minutes at 16,000 RPM. And in the second method, we can use mini bead beater apparatus. For example, for those isolates and for those bacteria or lactic acid bacteria which have a stronger cell wall, so we cannot isolate the DNA with the help of heating, we can use mini bead beater apparatus in order to disrupt the cell wall of lactic acid bacteria and extract the DNA from the cell, from the bacterial cell. آقای رضوی من تو جلسه هستم. بله.
خاموشی اگه امکانه داشت یک زنگ به من بزنی یا تلفن شو برای من بفرستی.
چشم متشکرم.
ممنونم.
Okay, sorry, excuse me. Okay, do you have my slides? Do you have my slides?
Yes, doctor.
Okay.
Yes.
Okay, let's continue with the next slide here. We have a PCR amplification of 16S rRNA gene. In order to perform a PCR, we should have one pairs of primer. I mean, we have one reverse primer. We should have reverse primer, and also we should have forward primer. And here the reverse primer is 4092R or reverse, and 27F, which stands for forward primer. This is the forward primer, and this is the reverse primer. And here we have the PCR reaction. As you can see, these are the PCR components or PCR ingredients, which is composed of water, a master mix, DNA, and primer. And these are the volumes or the amounts of each component in the PCR reaction. And we should perform the PCR and amplification of 16S rRNA gene with these two primers. Okay. In the next slide, you can see the thermal program. This slide shows the thermal program of amplification of 16S rRNA gene. As you can see, this is the picture of thermocycler, and we can perform and start, and we can run the PCR in this apparatus, in this equipment, in the thermocycler. And as you can see, the first step is the denaturation step, the initial denaturation, which is started at 95 degrees centigrade for 5 minutes for one cycle. After that, in the second step, we have again a denaturation step, and after denaturation, we have a annealing step at, for example, 50 degrees centigrade, and after annealing, we have the extension. After extension, we have final extension at 72 degrees centigrade for 10 minutes, and finally, we can store the PCR products or amplicon at 4 degrees centigrade. And this part can be repeated for 30 to 40 cycles. Okay, after performing the PCR, we have the gel electrophoresis, and we should perform the electrophoresis. Agarose gel, we should prepare the agarose gel in TAE or TBE buffer 1%, and the electrophoresis conditions are 9 defined voltage or volts, 35 minutes, and finally we can see the picture of gels like here in the gel documentation equipment. We have here in the gel documentation, and finally we have a picture like this picture, which shows the bands, the different bands, and these bands represents the DNA of each isolate here. In the next step, after finishing the PCR, we should use the amplified ribosomal DNA restriction analysis, or ARDRA. ARDRA, why we should use these techniques? Because in some cases we have many isolates, I mean many lactic acid bacteria or many bacteria, which is extracted from the plates, which is collected from the plates, I mean like MRS or M17. For example, if we have 100, 200s, for example, isolates, before sending these isolates to sequencing, it is better to perform ARDRA technique, because ARDRA, this technique decreases and reduces the number of isolates, and we can save my time and or money also with this technique, I mean with the ARDRA. So in the ARDRA, as you can see, amplicons were subjected to ARDRA by digestion with the restriction enzymes, and here with two popular and known enzymes, HI3 and HHA1, which these two enzymes is used for lactic acid bacteria. Okay, DNA fragments were separated by electrophoresis in 1.5% agarose gels at 75 volts for 90 minutes. And in the next step, after performing the ARDRA, now we can use representative amplicons, as you can see here, representative amplicons of different ARDRA profiles were sequenced using the primer 27F or forward primer. In order to sequencing and sequence comparison, we should send or representative amplicons or PCR products of different ARDRA profiles to sequencing. On average, here we have 850 base pairs were obtained per sequence, and the sequences were compared. In the next slide, as you can see, we have here a picture. Now here we have a picture from the sequencing, and we should editing the sequences using BioEdit and Chroma software. Here, as you can see, we should edit the sequences using these two softwares. We have these two bioinformatics softwares like BioEdit and Chromos. These are two softwares, and we should edit the sequences. And after editing, sequences showing 97% similarities or higher were deemed to belong to the same species. This percent is very, very important. This is like a cutoff. For example, if the similarity is 97 or more than 97, it is okay. And after comparison in the NCBI or gene bank, we can identify the lactic acid bacteria. For example, if it is assigned to Lactobacillus plantarum with the 97% similarity or higher, we can say that, okay, this bacteria is identified as Lactobacillus plantarum, for example. In the next step, after sequencing, we have another typing method, which is called Rapid Pcr or repetitive PCR. And this technique is used for typing of isolate using Rapid Pcr. And as you can see, we have only here one primer, which is called Basrah A2R. This primer is used for Rapid Pcr. And this table shows the component of reaction, DNA, master mix, primer, and sigma water. And these are the volumes of each components, of each ingredients in the PCR. And this is the thermal program and profile in Rapid Pcr. And as you can see here, again, we have one step of denaturation, and again, denaturation and annealing at 40 degrees centigrade. And after annealing, we have extension. And after extension, we have final extension, and finally we have a 4 degrees centigrade. For Rapid Pcr, we have a gel condition as following, as you can see, agarose with the 1.5% agarose gel, and time of electrophoresis is 90 minutes at 75 voltage. Important note here, there is an important note here, and this important note is that all isolates should have the same DNA extraction methods for Rapid Pcr. In order to perform the Rapid Pcr, all isolates should be extracted with the same DNA extraction method, and this is very important. And here in the next slide, you can see the different steps of Rapid Pcr, different steps of this procedure, of this method. As you can see here, first of all, bands profile at the end of analysis. At the end of Rapid Pcr, you will have a picture like this, and these bands represents the different patterns, as you can see here. And these are different isolates, which is run at the PCR, and you can see this picture. So we should convert and we should change these bands into some codes, I mean ASCII codes in Excel, convert to 0 and 1 codes in Excel. For example, every point which there is a band, we should put 1, and every position that there is no band, we should put 0 for that position. So we can change and we can convert these bands into 0 and 1 codes in an Excel file. After that, we should convert and transfer Excel file to the MVSP, transfer to the software of MVSP version 3. MVSP stands for multivariate statistical package, and this is the statistical package which is used for this technique, I mean for Rapid Pcr technique. In the next step, similarity content of patterns using simple matching coefficient, and in the next step, clustering of bands patterns using the UPGMR on weight per group method with arithmetic mean. And finally, you have a dendrogram here, like this dendrogram. And according to this dendrogram, you can type and you can differentiate the isolates. Okay. In the next slide, let's come back to the Dgge. As you can remember, in the first part of my lecture, I explained for you about a Dgge. And here again, we come back to the mechanism of culture-independent technique, or Dgge. And here, this is the picture of denaturant gradient gel electrophoresis, or Dgge, and this is the gel. This is the picture of the gel. And let's start with the gel. As you can see, this is the denaturant concentration, which is increases from 0% to 100%, and this shows the increasing of concentration of denaturant. And the denaturant here is a polyacrylamide. Polyacrylamide is a denaturant agent here, and the concentration is increased from this 0% to 100% here. And again here, this is the electrophoresis from top to bottom, and this is the direction. This is the direction of electrophoresis condition, or electrophoresis current, from top to bottom here, and this is the direction of electrophoresis. And here, as you can see, at the start, we have a double-strand DNA. Here, we have a double-stranded DNA at the start of Dgge. And during the Dgge, we have partially melted mutant and partially melted wild type. And also, we have two types of microorganisms, or two types of DNA here, and this symbol which shows the wild type, and this symbol which shows the mutant type of microorganisms. During the electrophoresis, or during the Dgge, as you can see, the two strands of DNA are melted gradually, little by little, and finally, at the end of Dgge here, the two strands of DNA completely separated, and is converted to single strands. And this is the mechanism of culture-independent technique, or Dgge. In Dgge analysis technique, we should have a DNA extraction from samples of milk, curd, or cheese, as I told you before. We should extract the DNA. Directly from the food samples, like milk, curd, or cheese. And here we can use from some kits like Kia AMP DNA stool mini kit, or QIAGEN, Germany. And after DNA extraction, again, we have a measurement of quality and quantity of DNA using nanodrop spectrophotometer. And after that, amplification of 16 srRNA gene with the help of following primers. And here again, we have one forward primer, F357 GC-clamp, and one reverse primer, R518 primer, and these are two primers which is used for 16 srRNA gene amplification in the Dgge. And in the next slide, as you can see, this is the Dgge apparatus. the Dgge is performed and is done in this apparatus, in this equipment, and this is the equipment of Dgge, as you can see, and electrophoresis condition here, and this is the table which shows again a component of reaction here, DNA, master mix, primer, and sigma water, and here again we have a thermal program of Dgge. After performing the PCR, the PCR Dgge, again we should have electrophoresis condition, electrophoresis conditions in Dgge. In Dgge, we have a polyacrylamide gel, and we should prepare a polyacrylamide gel, as I explained for you before, in decode or biorad apparatus at 60 degrees centigrade. And the best and optimum condition for separation for bacteria, as I told you before, at 40 to 60% concentration of denaturing gradient, and the denaturing agent is polyacrylamide here.
Please change your slide, please.
Yes. It's okay, and now it's okay, doctor?
Okay.
Okay. And here is the electrophoresis condition of Dgge in polyacrylamide gel in decode or biorad apparatus as at 60 degrees centigrade. And the best and optimum condition for separation of bacteria. For example, if we want to identify the bacteria or prokaryotic cell like lactic acid bacteria, the best and optimum condition is at 40 to 60% denaturing gradient. And this table shows the conditions of electrophoresis for bacteria, of course. Time of electrophoresis is adjusted to 16 hours. Electrophoresis voltage should be adjusted at 75 voltage, and denaturing gradient should be adjusted at 40 to 60%. And finally, in the last slide, as you can see here, this slide shows the bands identification in Dgge. As you can see here, these ladders shows the control or shows the marker. As you can see, this is the marker B, which includes or which compose of Enterococcus faecium, Enterococcus fecalis, Enterococcus faecium, Enterococcus fecalis, Lactococcus lactis, Escherichia coli, and Lactobacillus paracasei from top to bottom. And this is the marker B. And here is a marker A. And marker A, again, we have 4 bands, which is representative for these bacteria, Lactococcus garviae, Lactobacillus plantarum, the second one. The third one is related to Leuconostoc mesenteroides, and the fourth one is related to Streptococcus parauberis. And these ladders and these controls help us to identify exactly these bands. And after showing these bands, as you can see, these bands, and these bands related to marker A, these bands related to marker B or control. And these bands, we should cut the bands, and after cutting the bands from the gel, we should put the bands in a distilled water for 24 hours. And after that, re-amplification using the primers, I mean F forward and reverse primer, but here without GC clamp. For the re-amplification, we use these forward and reverse primers, but without GC clamp. And finally, sequencing and comparison of sequences. After sending the PCR product and the bands to the sequencing centers, the sequencer centers will send us the results of sequencing, and we can use the comparison. We can use the comparison with the NCBI and gene bank, and we can identify the isolate like lactic acid bacteria with the NCBI or with the gene bank. And here in this slide, my lecture is finished. Thank you very much for your attendance and for your attention to my lecture and to my presentation. And here I am ready. If you have any question, I am ready for your answering to your question. Thank you very much again. Thank you for your attention. The doctor, do you have my voice? Can you hear? Dear doctor? Dear professor, can you hear me? Can you hear me? Can you hear me? Dear professor, can you hear me? If you have my voice, please reply to my voice, because I don't have any voice. Can you hear me, dear professor?
Yes. Yes, doctor.
Huda Aboud, yes, you are?
Yes, yes.
Huda Aboud?
Yes.
Oh, okay, can you hear me?
Yes.
Okay, but I can't I can't hear the professor, the visiting professor, I mean. Maybe the connection was lost, yes?
Okay,
Yes.
thank you. No, I went to your lecture.
Okay, thank you, visiting professor. Can you hear me?
Yes, I can.
Okay, okay. And my lecture was finished, and now I am ready for your question. If you have any questions, I am ready for answering to your questions.
طلاب لأي واحد عنده سؤال للدكتور البروف يمكن يسأل الآن عن طريق الميكروفون. أكو عنده سؤال طلاب أساتذتنا؟
No, doctor, not any questions.
Okay.
If you have any question, I am ready for your question.
Yes, Zainab Nahi with you.
No, doctor, no any questions.
Thank you.
Very interesting.
Yes.
Thank you.
Okay, doctor, thank you
very much.
so much for your lecture, and we are interested in the slides, and you give us some more information, and thank you so much.
Thank you.
And
Thank you very much. I hope this lecture will be suitable for you and for your audience and for your students. I hope my lecture will be useful and will be suitable for all students from Basrah University.
I hope to meeting you another time, inshallah.
Thank you very much, inshallah. في أمان الله. في أمان الله. خدا حافظ. خدا حافظ.
الطلاب يقدرون تغادرون المنصة إذا تحبون. انتهت المحاضرة.